Human anatomy and physiology. Here we are in episode 3 on the urinary system and fluid, electrolyte, and acid base balance. In this particular episode, we are focused in on the regulation of water in the human body. As we've spoke about previously, the human body consists primarily of water. There are two main compartments with the intracellular fluid and the extracellular fluid. On this particular slide now, we are looking at the regulation of both water and electrolyte levels in the intracellular fluid. What you can see here is that there are tight regulation of both water and electrolyte levels. Regulation is controlled mainly by mechanisms that regulate the movement of solutes in water between the extracellular fluid and the intracellular fluid. Notably on the screen here, you can see that lipid bilayer, also known as a plasma membrane, and that's what separates out both the intracellular fluid and the extracellular fluid compartments, and thereby can regulate their fluid composition. You can also note that there is a movement of solutes across the membrane, and to accomplish this, there are pores or ion channels and active transport mechanisms that allow for ions such as sodium and potassium to move across the membrane, while at the same time preventing larger proteins from doing so. Notably, an unequal concentration of electrolytes on either side of a cell membrane will create an electrical charge difference. On this next slide, we look at the regulation of water and electrolytes in the intracellular fluid, which depends on electrolyte balance and fluid balance. And so an imbalance in fluid balance will cause an imbalance in electrolyte balance. Here what you can see, if we follow this flow chart, is that when you have a change in the concentration of sodium ions in the intracellular fluid, such as a decrease, this has an immediate impact on the interstitial fluid osmotic pressure, which will also decrease. As a result, it becomes hypotonic compared to the osmotic pressure in the intracellular fluid. As a result, water will move by the process of osmosis from the interstitial fluid into the intracellular fluid. I'm going to jump forward now to look at another diagram which highlights the extracellular fluid and the intracellular fluid and how the plasma membrane has pumps that facilitate the exchange of sodium and potassium. The sodium-potassium pump is shown here. It requires ATP and the breakdown of ATP into adenosine diphosphate and inorganic phosphate. And what this does is it is going to allow for the transfer of sodium out of the cytoplasm and into the extracellular fluid. What you'll also notice is that the pump can transfer potassium out of the extracellular fluid and into the cytoplasm. Although sodium and potassium can leak through pores into and out of cells respectively, the high levels of potassium and low levels of sodium in the intracellular fluid are maintained by these sodium-potassium pumps in the cell membrane. Once again, I'd like to highlight that this is a form of active transport, as these pumps use energy supplied by the breakdown of ATP to pump sodium out of the cell and to allow potassium into the cell. This next chart highlights 4 types of pressure that really modulate fluid and electrolyte levels in the plasma and interstitial fluid and how fluids will shift. I've defined all 4 of these types of pressure at the bottom of your screen, but essentially we have interstitial fluid hydrostatic pressure, which is generated by the water in the interstitial fluid itself. We have blood hydrostatic pressure, which is generated by the water that's inside of the capillaries that makes up the proportion, a proportion of blood. And as we've discussed in a preceding lecture episode, the hydrostatic pressure within a vessel is highest at the arterial end when we're talking about capillary exchange. We also have blood colloid osmotic pressure, and that is generated largely by plasma proteins and retains water like a sponge. We gave the example of the leaking bucket that has 1 sponge versus has 10 sponges, and how the bucket that has more sponges will retain more water. This is akin, or it's an analogy to this osmotic pressure. We also have interstitial fluid colloid osmotic pressure, and this usually sits on close to 0 millimeters of mercury, and that's due to a lack of proteins that is typically within the interstitial fluid. So if we take each of these 4 pressures and we look at comparing the sum of the hydrostatic pressures and the osmotic pressures, you can see that when the blood hydrostatic pressure combined with the interstitial fluid colloid osmotic pressure, when those 2 are summed together and are equal to that of the interstitial fluid hydrostatic pressure and the blood colloid osmotic pressure, then in fact you have no transfer of water between these compartments. When they do not equal each other, that is in fact when there's a net transfer of fluid. In fact, when the blood hydrostatic pressure and the interstitial fluid colloid osmotic pressure are summed together and they are greater than the other 2 types of pressure, then in fact fluid will shift out of the blood into the interstitial fluid. In contrast, when the blood hydrostatic pressure and the interstitial fluid colloid osmotic pressure are less than that of the interstitial fluid hydrostatic pressure and the blood colloid osmotic pressure, that is when fluid will shift out of the interstitial fluid into the blood. This is similar to what we observe in the kidneys with the movement of fluid that exists, and we've spoke about previously. So that brings us now to this next example, which just summarizes what we've talked about in the preceding slides. And we've also highlighted this in a preceding lecture episode. Once again, when we're at the arterial end of a capillary and we're looking at the capillary exchange and the likelihood of fluid moving out of or into the capillaries, what happens is that as blood flows through the arterial end of the capillary, due to a high hydrostatic pressure within the blood, fluid will tend to move out of the capillaries. You will recall that the interstitial fluid colloid osmotic pressure tends to be quite low, close to 0, and therefore it's really relying on that BHP to be very high, or that blood hydrostatic pressure. A very high blood hydrostatic pressure at the arterial end of the capillaries, it was what is going to facilitate the movement of, once again, movement of fluid out of the capillaries. On the opposing end, when we're looking at the venous end of the capillary, so blood has flown through, we've had some level of capillary exchange, it equalizes out. At the venous end of the capillaries, in fact, the blood hydrostatic pressure tends to be much lower, and the osmotic pressure now tends to be higher. In this case, what you see is that the fluid will move into the capillaries. And this last slide really just highlights a clinical example. So what we have here is edema, which is a classic example of fluid imbalance, and in this case has occurred due to an allergy. This may occur in any organ or tissue, but common locations include the lungs, brain, and lower back. And this a possible cause of edema may be a decrease in the osmotic pressure within the blood due to a decreased concentration of plasma proteins. You may also have edema when you have an increase in capillary blood pressure or that blood hydrostatic pressure due to the venous congestion of heart failure. Lastly, if you have a retention of electrolytes, especially sodium, in the interstitial fluid, this can also lead to edema, and this may be caused by an increase in aldosterone secretion or kidney disease.